Okay, good morning to everyone. Today topic is microbes in human welfare. The microbes, it is derived from Greek word. It is called a small. It is called a small. Microbes is derived from Greek word. It is called as small. Means small microorganisms. Small microorganisms. So the small microorganisms are two types. One is useful microorganisms. Second one is harmful microorganisms. Harmful microorganisms. Now we are seeing useful microorganisms. The first 1674 Anton von Leeuwenhoek is discovered in first microorganisms. Anton von Leeuwenhoek discovered living cell. Living cell, where the living cell is conducted their experiment in bacteriology. I mean bacteria. Bacteria. 1674 Anton von Leeuwenhoek, he is conducted the experiment in bacteria. So he also observed a living cell first in bacteria. So he is a father of microbiology. Father of microbiology. Microbiology deals with microorganisms. Okay, he is a father of microbiology. Microbiology deals with microorganisms. So he is a father of microbiology. Microbiology deals with microorganisms. So they are present in everywhere. Means soil, water, air, plants, human beings. The microorganisms is everywhere in the universe. Means, so they are omnipresent organisms. Omnipresent organisms. Because why they are called omnipresent organisms means they are invisibly to feel in the everywhere in the universe. So we are not observing the microorganisms because with the help of microscope only we have to observing the microorganisms, because they are very small microorganisms. Okay, so they are present in everywhere in the universe. Okay, soil, water, air, plants, human beings. Okay, that's why we are calling omnipresent organisms. Omnipresent organisms means invisibility to feel in the everywhere in the universe. Okay, they are for example, they are cultured in our laboratory in only visible for bacterial cell and fungal cell. Bacterial and fungal colony. Bacterial colony and fungal colony. So in our laboratory, So we have to culture these type of microorganisms to only to culture and to visible. So we have to see the diagram in bacterial colony and fungal colony. Bacterial colony, fungal colony. Bacterial colony, it is to be displayed in this manner. It is to be displayed in this manner. In fungal colony, so in our laboratory, so displayed in these two colonies. Okay, so now we are moving the microorganisms. So what are the useful microorganisms in the human welfare? How it is to useful? So what are they? So we are now discussing. First, the useful microorganisms are, the first thing is household products. Household products. How to useful microorganisms in household products. The first, in houses, first curd. Commonly we have to see in our houses in the curd. So how to convert curd? The microorganisms are how to help to convert curd. Okay, we have to take fresh milk. We have to take fresh milk to add inoculum or starter or some amount of curd. Inoculum or starter or some amount of curd. Okay, in the curd is having lactic acid bacteria. Lactic acid bacteria is converted to curd. The lactic acid bacteria releases lactic acid. In this lactic acid, partially digest milk protein. Partially digest milk protein. In this lactic acid, partially digest milk protein to convert curd. Okay, the fresh milk, we have to take the fresh milk to add inoculum or starter. Okay, that is having lactic acid bacteria. That lactic acid bacteria is releases lactic acid. That lactic acid partially digests the milk protein to convert curd. So at the same time, lactic acid bacteria releases B12 vitamin. It is to useful in our body to kill pathogens. Okay, the fresh milk is having milk protein. So the lactic acid is partially to digest the milk protein to convert curd. And second thing is dough. Dough is having bacteria. This bacteria, it is highly to release carbon dioxide. Highly to release carbon dioxide. So the carbon dioxide, it is to react in our dough. It is to react in our dough. It is react in our dough. It is given a bulgem or bubble shape. Bulgem or bubble shape. Bulgem or bubble shape means the bacteria will be grow. The bacteria will be grow in our dough. Grow in our dough and releases carbon dioxide. So releases carbon dioxide. After one day, it is available to prepare idli or dosa. Idli or dosa. Idli or dosa. Idli or dosa. It is a second thing. Dough means, so it is one of the useful idli or dosa. It is one of the useful idli or dosa. So a dough is having bacteria to grow bacteria to release carbon dioxide. After dough it is useful for idli or dosa. Idli or dosa. It is a second household product. First one is curd. Second one is dough. Next one is baker's industry. Baker's industry means bread. Okay, baker's industry means bread. Bread means here yeast. Yeast means Saccharomyces cerevisiae. Saccharomyces cerevisiae. In this organism is to be converted into bread. So one more thing is taddy. You know taddy? Taddy means most South Indian peoples will drink. In this taddy. More South Indian peoples will drink this taddy. In this taddy is having 6% alcohol. Taddy produces palm plant sap. Okay, taddy means, so most South Indian people will drink taddy. In this taddy is having 6% alcohol. So the taddy will be produces palm plant sap. Okay, the first thing is curd, second one is dough, third one is bread, fourth one is taddy drink, and one more thing is cheese. You know cheese means it is one of the dairy product. Okay, so the amount of microorganisms will be added. The cheese will be given a different flavors. For example, you have to add 10% of microorganisms, so it will be given a one flavor of cheese. So 20% of microorganisms, 30% of 40, 50, 100. So you have to add different types of amount the microorganisms. Okay, microorganisms will be added. It will be given different types of cheese flavors. Okay, the first one is Swiss cheese. Swiss cheese. First one is Swiss cheese. So it is to be preparing for Propionibacterium sharmalae. Propionibacterium sharmalae. Okay, the Swiss cheese will be prepared Propionibacterium sharmalae. Propionibacterium sharmalae. So high amount of CO2 will be released. High amount of CO2 will be releases. High amount of CO2 will be releases. So the flour is called Swiss cheese. The flour is called Swiss cheese. Swiss cheese. Next, Roquefort cheese. Roquefort cheese. Roquefort cheese. Roquefort cheese. To add fungus or fungi. To add fungus or fungi, it is given different types of flowers. Different types of flowers in the cheese. In the cheese. It is one of the dairy product. Dairy product. Cheese is the one of the dairy product. So you have to add a different percentage of microorganisms will be added. Means the amount of microorganisms will be added. It will be given different types of cheese. Different types of cheese. Swiss cheese, Roquefort cheese. Roquefort cheese, and different types of cheeses will be come. Different types of cheeses will be come. Next, the first household product is completed. Household product is completed. The first one is, what is that? Curd. Curd will be milk will be converted into curd. Milk will be converted into curd. Second thing is dough. Okay, dough is having bacteria. That bacteria will be grow and releases carbon dioxide. After, the dough will be very useful to idli or dosa. It is useful to idli or dosa. Next, baker's industry means bread. The yeast or Saccharomyces cerevisiae, it is to be converted into the bread. Next one is toddy milk. That is most South Indian people will be drink. Toddy. Means drink toddy drink. That is having 6% of alcohol. They are producers from palm plant sap. Okay, next thing is cheese. It is one of the dairy product. So we are adding different amount of microorganisms to be given different types of flowers. So household products is completed. Next thing is called industrial products. Industrial products. The first one is household products. It is completed. The second thing is industrial products. The industrial products produces beverages and antibiotics. Okay, antibiotics. Okay, we are using in beverages and antibiotics in preparation. We are using fermenters. Okay, the industrial products produces beverages and antibiotics. In that produces, we are using fermenters. Fermenters means it is used to store microorganisms in large amount. Okay, means fermenter means large containers or large vessels, because of they are storing microorganisms. They are storing large amount of microorganisms. Also, it will be developed on the large containers or large vessels. That is are calling fermenters. That is are calling fermenters. Next, in these beverages and antibiotics, the first producers is called ethanol. The first producers is called ethanol. Ethanol preparation. The first ethanol preparation, ethanol means it is also one of the alcohol. It is one of the alcohol. To produce us, which particles are to use to prepare ethanol? We have to see here. Okay, malted cereals plus fruit juices. To add yeast. To add yeast means Saccharomyces cerevisiae. Saccharomyces cerevisiae microorganisms we are adding in this bowl to convert ethanol. Means the ethanol production is called using materials are malted cereals plus fruit juice, adding Saccharomyces cerevisiae microorganisms to add yeast or fungi, yeast membrane, to add Saccharomyces cerevisiae to produce ethanol. Next, in this alcohol will be produced in different types. The first means beer and brandy, whiskey, rum and wine. So beer and wine are producing in this beer and wine and brandy, whiskey, rum. So they are producing industrial products. These all are also called alcohol. Alcohol products, and ethanol also alcohol products. And wine and beer, it is to be producing without distillation. Means with distillation. In these three things called without distillation. Wine and beer produces fully with distillation. Brandy, whiskey, rum without distillation. Okay, without distillation. Industrial products produces beverages and antibiotics. Beverages means it is production of alcohol drinks. The first production is called ethanol. So what are the raw material? Malted cereals and fruit juice to add Saccharomyces cerevisiae to produce ethanol. And wine, beer, brandy, whiskey, rum. So also one of the production of beverages. And wine and beer with distillation, brandy, whiskey, rum without distillation, the beverage productions. And next coming to antibiotics. The first antibiotic is called penicillium. It is coming from Penicillium notatum. Penicillium notatum. Penicillium notatum. Penicillium notatum. Penicillium notatum. So who is discovered? The Alexander Fleming. Alexander Fleming. Alexander Fleming. Alexander Fleming is 1928. 1928. He is conducted one experiment. He is conducted one experiment in petriculate. In petriculate. In petriculate. Conducted experiment using the microorganism is called Streptococcus. 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 Okay, he is conducted. Alexander Fleming is conducted the experiment in 1928 in which organism? Using which organism? Streptococcus. So he is observing on in the petriculate, the Streptococcus organism is not growing, means not developed in the petriculate. Not developed in petriculate. He is observing on the petriculate. He is observing on petriculate. At the same time, he is also observing some amount of chemical. Some amount of chemical will be released in this Streptococcus microorganisms. So there was not developed on the petriculate Streptococcus microorganism. At the same time, they are releasing some much amount of chemicals. Some much amount of chemicals. Some much amount of chemicals. That chemicals is that chemical is observing. Then finally, that is called penicillium. That is called penicillium. Means finally it is converted to antibiotic. The name is given the antibiotic. Florian chain. Florian chain. Chain. Alexander Fleming was discovered penicillium. Alexander Fleming was discovered penicillium. So the name is given penicillium coming from Penicillium notatum. The is conducted the experiment is called Streptococcus. Streptococcus. But the name antibiotic, the name was given Florian chain. The fully converted into antibiotic. Fully converted into antibiotic. The Florian chain. Florian chain. The 1945. So the Nobel Prize will be awarded in these two scientists. Nobel Prize in 1945. Nobel Prize. The Nobel Prize awarded in these two scientists, Flori and Chain. Means they are fully converted into antibiotics. Means the name was given antibiotics. Means antibiotic means antibiotic means against life for pathogens. Against life for pathogens. In human beings, it is also useful, means pro-life. Pro-life. The antibiotics to against work of microorganisms, means to kill the microorganisms to useful human beings. To useful human beings. That's why the against life for microorganisms are pathogens. In human beings, pro-life means it is also useful. It is also useful. Okay, it is also useful. At the same time, the American soldiers in Second World War time. In American soldiers, Second World War time, these antibiotics are using. Second World War time, American soldiers also to be using antibiotic. This penicillium antibiotic. Penicillium antibiotic. Penicillium antibiotics. Next, coming to the chemicals and enzymes and bioactive molecules. Bioactive molecules. Chemicals, chemical producers, chemicals, enzymes, bioactive molecules. Bioactive molecules. First, chemicals. So what are the chemicals will be produced in the industrial? So what are the chemicals are produced in industrial level? Industrial level. First, Aspergillus niger. Aspergillus niger. Aspergillus niger to produce citric acid. Citric acid. It is also fungal membrane. Aspergillus niger bacterium. It is also coming from, it is a fungal membrane. So this bacteria means produces citric acid. Next, Acetobacter aceti. Acetobacter aceti. It is also bacteria. It is a bacteria. So it is produces acetic acid. Acetobacter aceti. It is a bacterium produces acetic acid. Next one is Clostridium butylicum. Third one is Clostridium butylicum. It is also bacteria to produces butylicum. Which acid? Butyric acid. Produces butyric acid. Okay. The first one is Aspergillus niger. It is a fungal membrane. This is produces citric acid. And second one is Acetobacter aceti. Is one of the bacteria. This is also to produce acetic acid. And Clostridium butylicum is one of the bacteria to produce butyric acid. First we have to see the chemical producers. The first Aspergillus niger, Acetobacter, and Clostridium butylicum. Next one is called lactobacillus. Next, fourth one is called lactobacillus. It is also to be producing lactic acid. It is also one of the bacteria. Next, fifth one. Already we know that the production of ethanol means called yeast, called Saccharomyces cerevisiae. To production produces ethanol. Okay, these are the chemical producers. Means first chemical producers means first thing is Aspergillus niger. So it is a fungal membrane to produce citric acid. Acetobacter aceti. So it is producing acetic acid. So Clostridium butylicum to produces butyric acid. And lactobacillus, it is to be producing lactic acid. So already we are discussing in this, means yeast, Saccharomyces cerevisiae, to produce ethanol production. Ethanol production. This is all our chemical producers. Chemical producers. Next coming to enzymes. 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 The first enzyme is lipase. First enzyme is lipase. Means the lipase is using to formation of detergent. Formation of detergents. Okay, the lipase enzyme using to formation of detergents. Like means different types of ring, surf excel. Okay, different types of detergents. Different types of detergents, and also to use removing for oil stains. Removing for oil stains. Oil stains, and also to use remove oil stains. All oil stains, and also using clots. And also using clots means clots is having cellulose enzyme. Cellulose is secreted by cellulose. Cellulose. Okay. Next enzyme is called proteases and pectinases. Next enzyme is called proteases, protease and pectinase. Pectase or pectinase. Okay. These enzymes are too useful. These enzymes are too useful bottle juices to use purifying bottle drinks. Bottled drinks or bottled drink juices, because of using for purifying. Okay. Proteases and pectinases are useful for bottle drink juices to purify. To store many days. Okay. These are the enzymes. These two are the enzymes. Okay. Next coming to bioactive molecules. Bioactive molecules means these two are lipase, protease, pectase. Means these two are the enzymes. The lipase is formation of detergents and also used to remove oil stains, and also using for clots. And protease and pectase, this enzyme also to be useful for bottle drink juices. To purifying, to store many days. Next coming to bioactive molecules. Bioactive molecules. The first one is streptokinase. Streptokinase is produced from Streptococcus bacterium. Okay, the streptokinase is producing from Streptococcus bacterium. This is to use myocardial patients. Myocardial patients means heart attack patient. The streptokinase is calling another name is called clot buster. Clot buster means the fat is to be clotted in the blood vessels. Okay, that's time to use streptokinase to removing clots, to removing clots. That's why these are calling clot buster. That's why this is calling clot buster. Means this is useful to myocardial patients. Myocardial patients. Myocardial patients are who is? They are heart attack patients. Means heart attack patients. Why heart attack will become means the main fat will be clotted on blood vessels. Fat will be clotted on blood vessels. Okay, so that's why heart attack will become. So the fat will be clotted on the blood vessels. It is to be removing for clotting. This is to removing for clotting. This is to removing for clotting. Next second thing is cyclosporin A. Cyclosporin A. Cyclosporin A. Cyclosporin A. So cyclosporin A is produced from Trichoderma polysporum. Trichoderma polysporum bacteria. Okay, cyclosporin A produced from Trichoderma polysporum. The cyclosporin is given to immunosuppressive. The cyclosporin is to be given immunosuppressive. Immunosuppressive means, for example, organ transplantation patients are there. Okay, in the organ transplantation time, then automatically immunity levels will be down. So in immunity levels will be down, organ transplantation is not possible. So that's why the cyclosporin A will be given to increase immunity levels to easily to transplant organ. At the same time, so they are using continuity. They are using to continuity of their life, because of the other organ will be entered in our body. So our antibodies are not accepted that organ. That's why we are using immunosuppressive tablets, because other organ will be entered in our body. So the antibodies are not accepted that organ. So we are lifelong to use this type of injections or tablets. Means in organ transplantation time will be given cyclosporin A to increase immunity levels to easily to transplant organ. Means it is using for organ transplantation. Patients. Organ transplant patients. And one more thing is there. So what is that? One more thing is, the third one is called Monoscus purpureus. Okay, so this is Monoscus and purpureus producing from yeast. This is to use in our body. Cholesterol reducing agent in our blood. Cholesterol reducing agent in our blood. Cholesterol reducing agent in our blood. The Monoscus purpureus, it is to be producing from yeast. This is using in our body to reducing cholesterol agent in our blood. It is reducing cholesterol agent in our blood. So this is bioactive molecules. Bioactive molecules. One is streptokinase, second one is cyclosporin, third one is monoscus purpurus. Monoscus purpurus. Okay, these three things are bioactive molecules. Bioactive molecules. And next, coming to the sewage treatment. Sewage treatment. Next thing is sewage treatment. Next thing is sewage treatment. Next thing is third, sewage treatment. Sewage treatment. Sewage treatment. Sewage treatment. Sewage treatment mainly using for waste waters, means for example drainage waters. Okay, some waste waters, they are to cleaning to release some rivers and some oceans. Okay, so we have for example in some cities or there you have to see in wherever you go there will be drainage water, some waste water is there. So wastewater is there. That wastewater to purifying to release rivers and canals and oceans. To release rivers and canals and oceans. So that is called sewage treatment. The sewage treatment are using to purifying the water to release, purifying the waste material to clear the water, to purifying to release canals and rivers and wherever there. Okay, it will be releasing also to some different types of canals and different types of oceans, small things are there. So there will be releases. There will be releases. Okay, now you have to see the treatment will be start in 3 stages. The treatment will be start in 3 stages. You have to see the 3 stages. Okay, sewage treatment will be starting 3 stages. 3 stages. It is called sewage. This is called primary treatment. Primary treatment, and this is called secondary, and this is called aerobic tank. Aerobic tank. Secondary treatment is having heavy machines or heavy fans is there in our treatments. Heavy machines means they are having different types of fans. Okay, the first thing is called sewage treatment. This is called primary treatment. Sewage, and that is called primary treatment. So primary treatment, it is called as a filtration. Filtration and sedimentation. Primary treatment is called filtration and also sedimentation. Filtration means what are the floated waste materials. For example, means like plastics. So what are the floated in water? What are the floated in water? Means some waste material will be floated on the water. Means some derbs. That is also called derbs. Okay, derbs. So floated materials and derbs will be filtered in this filtration process. Okay, so what are the floating materials in their water? What are the floating materials in their water? Means that is called the waste material will be floated. Waste material will be floated on the water. Means that is called some plastics, some derbs. Plastics and derbs. They are filtration process. They are removing for filtration process. And second thing is sedimentation. Second thing is called sedimentation process. Means it is called grit. It is to be filtration of grit. It is to be filtration of grit. Grit means it is a soil particles. It is a soil particles. Grit is a soil particles. Okay. Filtration process removing from floating materials and derbs, and sedimentation process to removing for grit and soil. Grit means it is one of the soil particles. Soil particles. After remaining particles is there, that is called effluent. So that is called effluent. Remaining particles will be going into the effluent. Remaining particles will be going into the another tank. That is called effluent. That is called liquid type. Effluent. Remaining particles are calling effluent. Remaining particles. Filtration, sedimentation after remaining particles. After remaining particles is called effluent. That is called a sludge. That is called a sludge. That is called sludge. Okay, that sludge will be going to secondary treatment. Okay, secondary treatment. Here secondary treatment. So what are the material? Sludge. Okay, the sludge will be coming into the secondary treatment. Secondary treatment means here the heavy machines are there. These machines will be continuously to rotate to produce air. Means the machines are continuously to rotate to produce air. So in the air to form oxygen. So that air is okay. Continuously the machines will be rotating. The air will be formed. So in the air, the oxygen will be there. Or formed. And in the oxygen here vigorous microorganisms will be developed. In oxygen means in the air. In vigorous microorganisms will be developed. Vigorous microorganisms will be developed. Vigorous microorganisms will be developed. Organisms will be developed. After develop to completely consume the sludge. The sludge will be coming the secondary treatment. So here the machines will be continuously to rotate to produce the air. So that air is having oxygen. So in their oxygen, the vigorous microorganisms will be produced or developed. Okay, wherever the form the oxygen, so the vigorous microorganisms will be developed. So developed microorganisms will be completely to consume the sludge. To completely consume the sludge to reduce oxygen. That means so completely to consume the sludge means whatever it is, oxygen also to there, oxygen also to be reduced means so the machines are continuously to rotate to produce oxygen. So I mean to produce air. To produce air, in the air oxygen is there. Oxygen is there. That's the time the vigorous microorganisms will be developed. Then the microorganisms, vigorous microorganisms will be developed. After develop, to completely to consume the sludge, to completely consume the sludge, and also to be reduce the oxygen, also to be reduce the oxygen, that is called biochemical oxygen demand. Biochemical oxygen demand, that is called BOD. That is called BOD. Biochemical oxygen demand. Biochemical oxygen demand. Biochemical oxygen demand. Biochemical oxygen demand. After I mean completely to consume the sludge, okay, the microorganisms will be reduces the oxygen means, okay, their biochemical oxygen demand, so after remaining will be going into the aerobic tank. Here also aerobic microorganisms are there. So here completely to consume sludge and oxygen. Here also there floc digesters. Here also there flocs digesters. Flocs digesters. Okay. So the remaining directly will be coming in aerobic tank. So in this aerobic tank is having anaerobic microorganisms here to present. Anaerobic microorganisms in present in this tank. Which tank? Aerobic tank is having anaerobic microorganisms. Here completely to digest or consume the sludge, that are called floc digesters. That floc digesters will be coming into the anaerobic tank here, and anaerobic microorganisms is there. Anaerobic microorganisms will be there in this floc digesters or floc digester. It is called other organisms will be digest. Okay, the floc digesters also to be digest. Reducing for anaerobic organisms to release biogas. It is called releasing methane. Okay, here this is floc digesters. The floc digesters directly coming in the aerobic tank. Aerobic tank is having anaerobic microorganisms. Anaerobic microorganisms will be floc digesters will be digest, then release methane. That is also called biogas. This is called sewage treatment. This is called sewage treatment. Okay. Remaining we are discussing tomorrow. Okay, thank you.